proteins All right, so we talked about amino acids, right, which led into the formation of peptides, and by the continuation of that, now we can talk about proteins. So proteins, whenever you have a whole lot of amino acids linked up together to form kind of this polymer of amino acids, which we're going to call a protein. So in looking at proteins, we can refer to their structures in different ways. So we're going to talk about their primary structure, secondary structure, tertiary structure, and even quaternary structure. So kind of 4 levels of structure of the proteins. So starting off with the primary structure of the protein. The primary structure is basically just going to be the sequence of amino acids. So remember that we identify amino acids based on their R group. So if you looked at the ball and stick model on the bottom of the page here, if you said that this first R group had a side chain that was a hydrogen, which would make it glycine, and you had the next one that had a side chain of CH3, which would make it alanine, and then the next one would have CH2OH, which would be serine. Let's say the last one was another glycine, just to be simple. You would say that this particular protein would have a sequence of glycine, alanine. So down here, let me write it this way: glycine, alanine, serine, and then glycine again. Right? So the primary structure is just the names of the amino acids in the order which they show up, coming from the N-terminus to the C-terminus. The secondary structure of a protein refers to localized regions of a protein. And what I mean by that is whenever you make a protein, it's not just a big long string. And this is usually where if I was in a classroom, I would grab a string or a cord and kind of wrap it. And you can almost wrap it into like a spiral type shape. If you make a spiral type shape with like a string, you're basically making what we're going to refer to as an alpha helix. So an alpha helix is a type of secondary structure that is important in the stability of a protein. So here's a picture of an alpha helix. So again, this is where you have a region of a protein that forms this particular structure. Alpha helices are held together by hydrogen bonds. So remember, hydrogen bonds are whenever you have a hydrogen that's bound to O, N, or F. So in proteins, we have H's that are bound to nitrogen. So we have NH's all over the place. So nitrogens are actually going to be in blue in this structure. So I'm going to point to a hydrogen right there. See that white or gray sphere? That's a H that's bound to an N. And it can hydrogen bond with something that can have a lone pair of electrons, usually an O, N, or F. And if you look at it, the O that's on this carbonyl group right here, so that's a C double bonded to O, is right next to that NH. So basically that NH's line up with to form a hydrogen bond, C double bond O. And if you look through this structure, you can find many, many different examples of that. So here's another one down here where you have an H lined up with an O. Right here you have an H and an O right next to each other. Here's an H and an O, right? So you'll see that these line up all the time in this alpha helix to form what we refer to as the secondary structure. Another type of secondary structure that's also held together by these hydrogen bonds is a beta pleated sheet. And in this case, the same thing, you can see the hydrogen bonds hold it together. So here's the H and an O, right? And an H and an O, and so on and so forth. So the idea is whenever a protein is made, it's going to fold up in the kind of the most energy efficient ways possible. And one of these such energy efficient ways is a beta pleated sheet. Another efficient way is an alpha helix. Now, the entire protein is not going to be all one big beta pleated sheet. or the entire protein is not going to be one big old alpha helix. So what we're doing is saying, I'm going to try to draw, right? So if you had a protein, this is going to be the N-terminus, and it goes like this, and then this region is going to be a helix, and then there's some other parts, right, like this, and then there's some other parts here, right? So maybe that is going to be the structure of a protein, where you have an N-terminus where I have an N, and then I have a C-terminus where I have a C. The alpha helix is going to be part of this structure that shows up right there. So that's going to be your alpha helix. So the secondary structure is part of the overall structure, but not the entire thing. And then over here, this part that I drew right there, that's going to be your beta sheet. So a protein can have an alpha helix or a beta sheet. Some proteins will have like 10 alpha helices and no beta sheet. They might have a whole bunch of beta sheets. So you can have any mixture of these particular secondary structures. All right, so as I mentioned, that most proteins have regions of alpha helices and beta sheets, and also regions that can't be characterized. These are often referred to as just a random region or the random coils. So whenever you look at the structures in your book, you'll see them kind of drawn with the symbols kind of shown at the bottom with the alpha helices and these kind of spiral looking green things, and the beta sheets with the arrows, and the random region is just the string looking structure. And the reason we use these ribbon diagrams is because the structure you see over here on the right, that's a lot more easy to kind of follow and understand than, say, a ball and stick model. And you can make ball and stick models, you know, from proteins like this. They're often just not as visually kind of obvious in terms of what it is you're looking at as the ribbon diagrams that kind of simplify the model. So a lot of times we're going to be looking at ribbon diagrams when we're looking at the structure of proteins. Okay, so the tertiary and quaternary structure of a protein. So if you think back to what I drew on the previous slide, and there's no way I'm going to be able to recreate that here, but if you had an N-terminus, and then you had an alpha helix, and you had some other stuff, and then you had kind of a beta sheet, and then you have a C-terminus. What we mean by the tertiary structure is the structure of that whole thing. So the tertiary structure is going to be the three-dimensional shape that is taken on by the entire polypeptide. And it forms in that tertiary structure. So the tertiary structure is pretty much determined by the amino acids and how they're able to fit next to each other to maximize all their interactions. So the second bullet point there says to maximize hydrogen bonding with water, nonpolar side chains are kind of forced into the interior of a structure. So if you imagine like a basketball or a soccer ball or some type of sphere like that, that was kind of really dense and compact, the nonpolar side chains are going to point to the middle of that ball to keep away from the outer part, which could come in contact with water, which is going to be on the outside, because again, most of these proteins are in a cell, and our cells are made out of water. So nonpolar things in the middle, and those are going to be stabilized by London forces because they're all nonpolar, right? Nonpolar things are going to have London forces with other nonpolar things. And then polar functional groups are going to be found on the outer edge of the structure because they can hydrogen bond not only with themselves but also with water that's in the solution that's going to surround the protein. Amino acids that have charged side chains, like the polar, or sorry, the basic and the acidic ones, right? Your amines and your carboxylic acids, the glutamic acid, the aspartic acid, the lysine, the arginine, the histidine. Those are your amino acid with charged side chains. So those ones are also going to be found on the outer edge of your protein, and they can actually interact with each other by what's called electrostatic interactions. And this basically means if you have an amino acid with a positive charge, a lysine, an arginine, or a histidine, that it can interact with something with a negative charge. An example of an electrostatic interaction would be, say for instance, a lysine, right? So lysine would be a positive charge, right? So that would be a lysine. interacting with something that's a negative charge, like a glutamic acid, GLU. Right? So those ones would get close to each other in proximity, and that electrostatic interaction would occur between those to hold it together. And the last type of interaction is the strongest of all of them, and that would be a disulfide bond. Disulfide bonds are the ones that occur between cysteines. So remember disulfide bonds we talked about previously, these occur between cysteine residues. And these are going to be the strongest of all of these because they are covalent. Right? Covalent is sharing electrons. All of the other ones we talked about here, the London forces, the hydrogen bonds, the electrostatic interaction, none of those are sharing electrons. Those are all non-covalent forces that hold things together. So disulfide bonds with their covalent bonds are going to be the strongest of these. So on this slide, the strength of the forces, the strength is going to increase going down. So the weakest are going to be the London forces, next comes the hydrogen bonds, and then comes the electrostatic interactions, and then finally the disulfide bonds. Okay, so here is a picture of a protein, right? Much neater and cleaner to see than the scribbles I've made on the previous couple slides, that show the N terminus over here where you have the NH3+, and the C terminus over here where you have that COO-, and then a whole bunch of stuff in between. And I just kind of want to walk you through the protein so you can see the different types of interactions. So in this first part here, we have some hydrogen bonding that can occur. So this would be like a beta sheet, right, that folds on each other, where you have these stacked hydrogen bonds kind of on these kind of anti-parallel strands, right? I mean anti-parallel because this one strand is going this way, the next strand is kind of going in that direction. But they're going to be held together by these hydrogen bonds, and that's going to be our beta sheet. All right, moving forward, next we're going to have just a random-ish kind of hydrogen bond. And the main point I want to make with this hydrogen bond is that this NH is drawn kind of in the backbone of the protein. And what I mean is in the backbone, it's not part of a side chain. It's part of that repeating unit, right? Remember our repeating unit in proteins is going to be NH CH with an R group, and then C double bond O, and then NH CH with an R group, C double bond O, so on and so forth. That NH is one of these. We can also sometimes have NHs in the R groups, and we'll look at that kind of in a different part over here. So for instance, actually right next to it, this hydrogen bond that I'm circling there, right? So notice here you have an H that is an NH that's in the side chain having a hydrogen bond. But this one is occurring within the backbone. So you have an NH in the backbone, a C double bond O that's in a backbone, right? Here's circling on the bottom is a C double bond O that's in the backbone. So that's where that hydrogen bond is being formed there. Now the hydrogen bond kind of next to it, right, would be an example of an NH that is in a side chain of amino acid that can also form a hydrogen bond. Next up in our list here, we have some London forces down here, right? So these are between nonpolar and neutral amino acids. So the side chains of these are nonpolar. Notice there's only carbons and hydrogens, so they're hydrocarbons. Then over here, kind of moving forward a little bit more, skipping past that other hydrogen bond that we already talked about. Here we have an electrostatic interaction, right? So that electrostatic interaction comes from a lysine, right? The lysine is going to be the positively charged one here, right? That has a plus charge, and then something next to it that has a negative charge. So the negative charge being either a glutamic acid or an aspartic acid, right? So those would be the two negative charges like the one you're seeing there. You can also have hydrogen bonds that occur between two side chains, which you see over here, right? So you have a serine and a threonine interacting with each other there. You have more London forces down here on the bottom, right? So again, some more nonpolar groups. And then the last one I want to point out is going to be the disulfide bond that you see here. And again, this is going to be between cysteine residues. So again, in terms of understanding your amino acids, like what should you know from your amino acids? You should be able to look at a protein structure like this and be able to pick out some of the amino acids. So for the London forces, I don't expect you to know the name of the one that are involved in these nonpolar ones. You should just know that those are going to be nonpolar. When it comes to an electrostatic interaction like this one here, though, you should be able to tell me which amino acids can form an electrostatic interaction, because you should know which ones can form a positive charge, lysine, arginine, and histidine, and which ones can form a negative charge, a glutamic acid or an aspartic acid. You should also know, in terms of the disulfide bond over here, which amino acid is involved in that, right? Cysteine. So those are the important things I expect for you to be able to know from your amino acids and looking at their structures. The last thing I want to point out on this structure before I click off of this slide, right, is the alpha helix here at the end, right? So this would be your alpha helix. So we have an alpha helix and a beta sheet, and then a whole bunch of other interactions that can stabilize this protein within its tertiary structure. So again, I focus on the tertiary structure there because we haven't talked about the quaternary structure yet. So a quaternary structure is whenever you have two or more polypeptide chains come together in one complex. So in other words, down here you have N, and then you have a whole bunch of stuff, and here's your protein, and then you have a C. And then you have a different protein over here that has an N, and it might go like this, and like this, and like this, and it kind of comes over here, and there's a C. And together, these two polypeptides, the orange polypeptide and the green polypeptide, come together to form a quaternary structure, which we would call a dimer. We would call this one a dimer because there's two things that come together. So dimer would be an example of the quaternary structure. And you can have a dimer, you can have a trimer where you have three different polypeptides coming together, and you can actually have up to hundreds of different polypeptide chains interacting with each other together to form a functional unit. So some of our enzymes that are involved in metabolism, for instance, are these really large complexes of proteins that all come together, that work together to help us with metabolism. There can literally be hundreds of different polypeptide chains that come together. A couple common proteins that you may have heard of that are involved that have a specific quaternary structure is insulin. So, right, insulin is two separate polypeptide chains that are held together by disulfide bonds. Again, disulfide bonds meaning cysteine residues. And then hemoglobin is a protein. Again, that's the one that carries oxygen through our body. And it's actually a tetramer in that it has 4 subunits held together. So again, a lot of proteins have these various tertiary and quaternary structures.